CLPP is caseinolytic peptidase P. It is expressed in response to mitochondrial stress, and it maintains mitochondrial hemostasis and proteostasis to preserve cellular functions. In this manuscript, we wanted to determine whether CLPP is required for reproductive competence and reproductive rejuvenation within the ovary. We used a global germline deletion of CLPP. It was a mouse model, and we found that CLPP knockout female mice were infertile. They made much less mature oocytes and less two-cell embryos, and they did not make any blastocysts. They also showed oocytes with spindle abnormalities. When we looked at their mitochondrial function, we found that their mitochondria had abnormal shape. They were smaller, more round, and they had any problem with fusion, and they had lesser expression of fusion genes. They had more mitochondrial stress, they had more reactive oxygen species, they had elevated mitochondrial DNA copy number, and they made less ATPs. We then checked whether the lack of CLPP caused earlier depletion of follicles in knockout ovaries. We found that by age 3 months, knockout mice had a 4-fold increase in atretic follicles, and by 6, 9, and 12 months, their number of follicles, the knockout mice number of follicles, were decreased, especially primordial and primary. We then assessed gene expression in the knockout mice at 3 and 6 months in oocytes compared to the wild type. We found that a number of pathways were affected, and especially the mTOR pathway, which has been implicated in aging. We then did an interesting experiment where we treated these mice with rapamycin in vivo and in vitro, and we were able to restore oocyte function at least partially. So our findings tell us that CLPP and mitochondrial stress response in general plays a role in reproductive competence and reproductive senescence, as CLPP depletion gives a phenotype consistent with accelerated depletion of ovarian follicles, which reminds us of ovarian aging.